For those of you who don't know me, I'm Rebecca Twist, and I'm the Humboldt Bay Mycological Society Fair Chair. And many of you also, if you're members of our club, will know me as the membership chair, because I greet you via email when you join online. So to start off our meeting tonight, we normally, this is the third Wednesday of the month. So this is not only a presentation that we have scheduled as part of our 2020 virtual mushroom fair, but this is also our monthly meeting, our regular monthly meeting. And we usually start our monthly meetings with a show and tell period between 7 and 7:30. And when we meet in person, people bring their mushrooms in and share what they've been able to find, and we all gather around and look at them and talk about what they are. So in lieu of that tonight, we thought that we would show a little bit of what we're seeing in our Blitz. For our virtual fair this year, we are having an iNaturalist Blitz. It started on this last Saturday, the 14th, and it will be running until this coming Sunday, the 22nd. This coming Sunday, the 22nd, is when we would normally have had our in-person fair. So this time period gives us a pretty accurate glimpse at the types of fungi that we would have been able to collect for our fair. And that lets us add that data to the data that we've collected for our fair for the last 42 years. So I'm going to give us a little overview of what we're seeing so far in the Blitz. If I can find it, I have so many screens open. No. Uh-oh, sorry. There we go. I apologize. Okay. So if you haven't visited our Blitz page yet, if you go to iNaturalist, you can search for Humboldt Bay Mycological Society, and it will show you our current Blitz project. And the main page here for the Blitz gives you an overview of how we're doing so far statistically. So we've had just over 1,000 observations posted so far. And out of those, there have been 265 distinct species that have been identified. So there may be more, but that we were only able to identify 2 genus at this point in time. So there are obviously a lot of species that are being shown more than once, because there's a lot of them out there. 100 different people have submitted those observations, and we have 59 people who've actively been involved in helping to do those identifications. So that this is sort of our dashboard that gives you that overview. And there is also a map. And for those of you who haven't used iNaturalist a lot, I'll just take a moment and show you how to find that. So from the dashboard, if I go here to, this is showing me thumbnails of the different observations in a grid. But if I choose map, then that's how I get into this. And I'm going to make that a full screen. And so this map right now has this highlighted area that is showing us the region that's been defined for our Blitz. So our Blitz has a geographical region, and it also has a time limit, so from the 14th to the 22nd. And our geographical region is all of Humboldt County, Humboldt County, Del Norte County, Trinity County, and the western portion of Siskiyou County that is west of Highway 3. And it's really interesting to see where our observations are coming from. So not surprisingly, a big bulk of our observations are coming from the greater Humboldt Bay area. So we're seeing down to Fortuna and up to about Oric here. And then we also have a concentration around Crescent City. And we're getting a few inland. This is Highway 299 that runs through here. So there's a few inland in on the way to Willow Creek. And we can also see where we need some more observations. So if you know somebody who's in Southern Humboldt, or if you're in Southern Humboldt, or if you're down in Southern Trinity County, or up in Eastern Del Norte, then please get out and take pictures of any mushrooms that you see, and help us to keep track of what's out there right now. Okay. And then I also wanted to take this time to show, let's see, here we go. These are the species that are most observed. This is another statistic that is available from that dashboard. And so you can see here the most observed species that we're seeing is Amanita muscaria. And there was a great question when Christian Schwartz gave his talk to us last Friday night about whether it's worth posting observations of things that are seen a lot. And Christian gave an unequivocal yes, it's worth it. And so if you see Amanita muscaria, keep posting it, even though you know that we've seen a lot of it. There is a lot of it out, and it's beautiful. So get pictures and post them. The false chanterelle, Hygrophilopsis aurantica, is the second place species that we're seeing a lot of. Lactarius deliciosus, Russula rhodocephala. We're seeing a lot of that along the dunes right now, and Stropharia ambiguus. So those are our top 5. And in the next row, we see Clitopulus prunulus, Agaricus subrudolescens, Boletus edulis, Hyphiloma capnoides, and Swillus tomentosus. And you could just keep scrolling down and seeing which ones are the top fungi that we're seeing a lot of. Okay. And then I have a few that I wanted, just specific observations that I wanted to highlight because I thought they were noteworthy. So I was excited to see this morning that Scott, our club president, who goes by Myco Dude, found a Bonartsevia. And this is one of the 10 fungi that Fundus is asking people to keep an eye out for. It's considered to be rare or infrequently observed. And so I was happy to see that Scott got to find that. And there's a map over here that shows where he found it. Also, I just thought this was a really nice picture. The observations that I've selected to share with you were the ones that I thought that the photographs really struck me. So Cataphilasma ventricosum is just such an impressive mushroom, and this is just such a textbook example of it and a really nice picture. I thought this was a lovely photo of Hygroscopy singeri, just the angle of it and the colors, how the redwood stuff and the clover, that's not clover, occidentalis, oxalis, that's the word. Just an absolutely gorgeous picture and really amazing to see one of the hygroscopies out already. We usually see those little later, and I mean, we're just starting to get macrofungi right now. So that was a gorgeous one to see pop up in the observations the other day. Robert Childs found this up at Prairie Creek State Park, and absolutely gorgeous specimen of Spirasus radicata, and he did a beautiful job capturing it, and he left it there. He was in the state park, and there's no picking allowed in the state park, but he did a really beautiful job of capturing what a gorgeous mushroom this is. And Humboldt fungi, I'm forgetting who this is off the top of my head, but he's a new member, and he is right up there among the top people in terms of number of observations that he's posted for our Myco Blitz. He's been out like every day scouring the landscape for mushrooms, and I just thought this was an absolutely gorgeous photograph. We have a ton of mycenas everywhere. There's lots of little fungi, and I just thought this Earth Star was gorgeous in there with all of those. And this is just, I don't know, this one just struck me. Really beautiful Cocora, Amanita calyptroderma. Beautiful job showing the different stages of development here and capturing, identifying features in just a totally aesthetically pleasing manner. So kudos to Mike Potts. And another one that just struck me because it was so beautiful, the Ampuloclytosobe clavipes. And it just, the lighting and the angle, and this captures that mushroom super well. The observer for this one went on to post a number of pictures along with in this observation that captured all of the important identifying features, but that picture just in and of itself kind of says it all. So those are my favorites, and we have a few other of our club officers who have also selected some observations that they would like to share with us. So let me stop that. Okay, there we go. Who's ready? Maria or Joanne, who would like to go next?
I'm super not ready. I just got off the phone with Scott, who can't get on Zoom anymore.
Oops.
We were trying to troubleshoot. So now he's not here and can't get on Zoom. And that's what I was doing for the past like 10 minutes.
Okay, well, we'll keep talking, and maybe you can help Scott while Joanne shows us a couple of the ones that she wanted to share. You ready?
I'm ready.
Okay.
I didn't have much time to prepare for this, and I can't say I had any of the aesthetic criteria that Rebecca had. I wanted to show you a couple of things that are so common, but perhaps overlooked. So here is my first one, I hope. Yes. Mycena purpureofusca is...
Okay, Joanne.
Yes.
Will you share your screen?
I'll try.
So at the bottom, it's a green, if you move your cursor down towards the bottom of the screen, there's a green button that says share screen.
No, there's not. I think I have to go back to Zoom to do that.
Yes, you need to be in Zoom.
Okay, hopefully I can find it. Oh, jeez.
And welcome, Scott. Glad to see that you're here.
I can't find it. The Zoom.
It's on the very bottom of your computer screen.
Yeah, but
there's a little. It's like a little camera icon.
I don't have that. I just have. I just have this learn how thing. I don't know where it went. Let's see. See what I can do.
Scott.
I'm trying. Be patient. I'm not real good at this.
Also, I just want to send out an apology and share this because there's somebody else named Scott who is in the attendees, and I just threw him into the panelist section.
Oh,
Then realized it wasn't Scott. Then demoted him back to attendee. So apologies out there to Scots everywhere.
well, was there a question while I was talking? I thought I saw a hand raised, but did anyone see if there was a question that came through? Rebecca Benson, maybe? No?
There was a raised hand, but then I didn't see a question attached to it.
Okay, okay.
So Rebecca, if you have a question and want to put it into the chat, there's a little like word bubble icon at the very bottom of your Zoom screen. You can click on that and type into it, and then we'll see it and be able to answer.
Rebecca?
Yeah.
So I have a box that says host has disabled share screen.
Oh. Let's see if Maria can fix that. So that means you
I
found
fixed
the
it.
share screen. That's good.
Well, I had to go all the way out and back in to join the thing, but I did find it, but now it won't let me do it. So
Well, I had made you a co-host, and then that should have worked. And then you weren't a co-host anymore. Now I've remade you a co-host.
okay. I don't know where I am now.
So now where it said this, the host disabled screen sharing, can you now see that you can share the screen?
Yes, I can. But it sent me into something entirely different with a whiteboard and launch Zoom meeting and Zoom cloud meeting.
Oh,
I have
yeah.
no idea.
So that's like all of the possible windows you can select. You want to select the window that has iNaturalist on it. There should be, if you have iNaturalist open in your computer at somewhere, then that should come up as one of your options for what to open, because it's giving you every option of what you could share for your screen.
Okay, can you see that?
No.
You'll have to go on. I don't know what's happened.
Well, I know
Oh,
Noah
okay.
has a few that he wanted to
Good.
talk about, so we could bounce over to Noah.
Good.
Okay. All right. Thanks for trying.
Sorry.
That's okay. All right, let's hear what Noah has to say. Noah is doing all of our identification. So he has looked at every single one of the observations that have been submitted so far.
Yeah. Good evening, everybody. So when I'm looking at this stuff, I mean, I glance at photographs, and a lot of the IDs I can do from thumbnail, but every once in a while I'll open up a photo and be like this, and I click on, yeah, it's old turkey tails in the log. But then if you keep zooming in, if you look, that's Rickenella schwartzii. There's no observation of that mushroom yet. So that's a species that someone can go out there and photograph and add it to the species list. We finally got Crinopellis pisceus, this little whitish one right here in the front. We finally got that added to the list today. So there's a bunch of species out there that you're not photographing that are growing, they're fruiting. I see them, they're in your photos. So do better, people. No, you've done well. A couple things I just wanted to bring up, kind of why I use the names I use, and that's all depending on the mood I'm in. So yes, we know we don't have Lecterius deliciosus in California, and there's 3 different species that I'm applying this name to that are showing up on, on this Myco-Blitz. There's this one here with the spruce, which is the one we've been calling Deliciosus variety pisceus. There's a Rantiosauridus, and there's a Deliciosus that's growing with the pine. So 3 different species of Deliciosus. for ease of just finding them and knowing them, I will just call them all Lactarius deliciosus. And if I'm in the mood to feel like typing, I'll type group or complex or something like that, or just mention which one it is. Aurantiosauridus, I can usually pick out from a photograph. It's a slightly smaller, duller colored one. So that's usually one that I can go see on a photograph and say, okay, that's it, but not always. Xericomelus. So some of you may know that Christian Schwartz, Jonathan Frank, and I screwed up your Xericomelus and gave you a bunch of new names for them. But when you get Zelleri versus actual purpurea, sometimes you can't tell them apart from a photo. And it seems like this is the area that fools me the most, kind of like those Xericomelus from Trinidad, Patrick's Point, Big Lagoon. I think they're Zelleri, and then I scope them and sequence them, and they're not, they're actual purpurea. So you have this population of actual purpureas there that have these pale lines around the outside margin of the cap, and they look an awful lot like Zelleri. Some of them may be. I would need a specimen to absolutely put a name on that. And then there's other things I just have no idea what they are. This thing here, I would like to have a specimen of this. So if you find something like this, please dry it and get it to me. And then there are things like this. I mean, I will go back. I have like a list of, it's getting larger, of about 50 things or so now that I got to go back to and decide what I'm finally going to call them. And so this thing here, it's like stuff like this bugs me because I've looked at this observation about 30 times now, trying to decide if I want to call it a Xericomus or Boletus smithii. And if I had, I mean, if I had the mushroom in hand, I would know in a second, but the photograph just doesn't show enough of the stipe. You know, with that cracking and stuff. So these are the things that keep me up at night. And since I'm in Massachusetts this year, I'm up late as it is already. So keep going out there, photograph lots of stuff, and you know, you're doing good.
Thank you, Noah. That's really helpful to hear from you about what you'd like to see more of, and that we should be taking pictures of all those little mushrooms that are surrounding the big mushrooms too. Let's see, Scott, did you have any that you wanted to talk about? Oh, you're muted, Scott. I don't know, can you unmute yourself?
Okay.
Oh, there we are. There you are. Okay.
Okay. I have a Crinopellus that's not loaded yet.
Ah.
And I love Schwartzii, but I haven't seen that one yet.
Okay.
Because that one you see all the time at Van Eck. It's a weed there. But I don't see it as much. Of course, I only did DR6. So, but I don't see it there. It grows in roads and pathways mostly. I think we're doing great so far. I'm very excited about how it's going. Not as many species, a lot of work.
Yeah. To those of you who are tuning in and aren't familiar with all of us at Humboldt Bay Mycological Society, Scott Davison is our club president, and Maria Morrow, who's listed as the Humboldt Bay Mycological Society on your Zoom screen, she is our vice president. And for those of you who have seen our t-shirt for this year, she is also the artist who did our t-shirt. And Joanne Olson is our program chair. So she is the person who got all of our speakers for the fair, and she gets all of our speakers for our monthly meetings as well. So that we are the club officers. So thank you all very much for joining us. So we have 5 minutes, and I'm loath to start a little early just because there may be people who are waiting until 7:30 to tune in. But I have a couple of announcements I can make unless there are any other. Okay, I'm seeing a question. Is the Blitz searchable on iNaturalist? Yes, so you can just search for Humboldt Bay Mycological Society. The name of our MycoBlitz project is Humboldt Bay Mycological Society MycoBlitz 2020. So, and I find that if by the time you type in Humboldt Bay Mycological, it comes up. There's nothing else that starts with those 3 words. Okay, any other? Kyle Sipes wants to know if Maria has shared her pics from iNat.
You mean from all of my inside time that I've been having? Or do you mean like my favorite pics?
I think
Because if you
maybe
mean my
he
favorite
means,
pics.
I think he means your favorite pics. Do you have any favorites?
I have like been going through and trying to follow Noah around in ID stuff. But I've definitely loved some things on there, but I went through and tried to find them before and then ended up dealing with some other stuff. And so I have done no picture finding, or I'll go through and look really quick.
Okay, sorry, Kyle, no faves. He says, alas. I also want to encourage as many people as possible to help with the identification for the MycoBlitz. It's pretty easy to do. And you don't have to live in our local area to help with identification. I know that we have a lot of people who are tuning in from out of the area. And so your observations of fungi in your area won't be listed in our MycoBlitz project, but you can participate by helping with identification. And even doing identification to genus is helpful. So I highly encourage people to participate in that way. And in fact, let me go back to, oh, here it is. I'm trying to share. I'll show one more thing here. So if you see an observation that you would like to help with identification, then underneath the picture is all of the activity regarding that ID. And so I have not yet participated in the ID on this. It seems like it's pretty well taken care of, but it doesn't matter how many people get involved. So the first person to suggest Ampuloclytoscopy clavipes was Mike Potts. And so I can choose to agree with that identification, and or I can come down here to, this is my window, and suggest an ID. So I could suggest something else, or I can write a comment. So I could just say, I absolutely love this picture, and the person who made the observation will receive that comment. So those are a couple of other ways that you can participate in our MycoBlitz, and I find it really rewarding to do that. I can also show you, while we have a moment here, at our MycoBlitz page, when you look at identifiers, there's a leaderboard here, and Noah is our designated identifier in terms of he is looking at every single observation. We also have Alan Rockefeller. He's doing this completely on a volunteer basis. He's just helping us out. He got interested, and he's in second place for identifications. This is Maria. She's been doing quite a few. I've been going in doing what I can. If you are new to mushrooms, this is a really fun way to learn a mushroom. So I just learned about Russula rhodocephala. And so every time I see someone submit a picture of a red Russula, I look and I see if it's Russula rhodocephala, and if it is, I suggest the ID. And I know Noah's going to come look at them, so if I'm wrong, he'll fix it. But it's a really excellent way to help yourself learn these identifications and to help you remember the names of things. So those are just a couple of other ways that you can be involved in our MycoBlitz, even if you do not live in the geographical area where we're doing the observations. Okay, so now it is almost 7:30. I'm going to make one final announcement before we introduce Noah, unless there's any comments or questions from the panelists. No, okay.
No, just that Joanne did mention that there was someone really interested in the bone Dartzeria occidentalis. And that individual commented to me about bone Dartzeria, as did several other people. So if you happen to see that one, apparently they're really excited about it, even though it's kind of like a weed where we live. So,
okay. All right, so one last announcement. Okay, so the talk tonight by Noah is part of our virtual mushroom fair this year. And we are having a series of presentations. And we've already heard from Christian, Sue Van Hook, Alyssa Allen, and tonight is Noah. But I wanted to make sure everyone on the Zoom presentation or on the live stream tonight also knows that Lee Von Durer is going to be speaking tomorrow night. He's a member of our club, and he has a business called Fungi Farm. He's going to be telling us about cultivating edible and medicinal mushrooms tomorrow night. And Chad Hyatt, who's the author of The Mushroom Hunter's Kitchen, is going to give us a cooking demonstration on Friday night. And we invite you all to join us for those as well. If you want the Zoom links for them, you can find those on our Facebook page. It's the Humboldt Bay Mycological Society on Facebook. Or you can go to our website. Our web address is HB for Humboldt Bay, mycologicalsociety.org. So hbmycologicalsociety.org, this is our homepage. And here under our calendar, there we go. All of the talks are on our calendar. So if you wanted to see Levon's talk tomorrow night, if you click on that, it opens a description and it includes the links. Okay, and the same thing is true for Chad's talk. Click on the talk and you'll find the links to the talk there. Okay, and with that said, I will now turn this over to Joanne, who's going to introduce Levon. Thank you all for coming. Not Levon, she's going, I just saw Levon's name. She is going to introduce Noah. Thank you.
Thank you, Rebecca. I think I'm unmuted. I think I've got everything clicked correctly this time. Most of you know Noah Siegel. He's been a member of our Mycological Society for quite a while. He has given many talks to us, and he has been an integral part of our mushroom fair for many years. No mushroom fair this year, but we have Noah again, which is great. Noah is co-author of the field guide, Mushrooms of the Redwood Coast. He's spent 2 decades photographing, identifying, and furthering his knowledge about macrofungi, traveling and lecturing on multiple continents as he follows the mushrooms. He's one of the premier mushroom photographers in the nation, with photos appearing on the covers and in articles of many journals, books, and websites, and winning numerous awards. I am so happy that Noah can be with us tonight for our mushroom virtual fair. Welcome, Noah.
Thank you, Joanne. I was trying to get in some last second INAT IDs there.
Go ahead if you want.
People may be like a little bit bored watching me ID. Big thank you to Joanne and HBMS. I mean, Joanne took me in as family when I first showed up 11 years ago, 12 years ago, passing through California, starting to do field work for mushrooms at Redwood Coast. And it's sad not being able to be there this year and being out mushrooming with her and seeing all you people. But such is life now, and I'm starting to get used to it. I've been home most of the year, and that's quite a change for me. But it's about 20 degrees outside right now, and there's no mushrooms, so kind of missing California right now. So just a brief warning. I have so-so internet at my house. And so if I disappear, I will be back shortly because I have backup plans. But when I gave a talk on Monday night, it wasn't an issue. So hopefully we'll get through this fine. And then a couple other things. Feel free to ask questions in the chat windows. We'll get to those at the end, and I can stick around and answer all those questions. So if you have questions through the talk, go ahead and ask them, and then I can easily go back to that section of the talk and talk more about that. That way I can just like sit here and talk to my computer, which is tough to get used to. So a lot of you, probably haven't noticed, the fact that I don't really look for above ground fungi that much anymore on the California coast. And part of the reason for that was there was a couple years where I went there and I could go the whole season and only find a handful of new things. So I was getting a little bit bored. But then I started going, I started looking underground because there's a whole new world of fungi underground. And so the title from this talk, it actually came from a text I got from Michael Castellano or Kaz. And he just sent me this text that said, "Stop walking on truffles." And it's the truth is that most of us are walking on truffles. When we're out looking for mushrooms, we're walking all over them. And once you start looking for them, you'll realize that there's incredible diversity of hypogeous fungi, of underground fungi. So to start so when I use the term truffles, I am talking about all hypogeous fungi. And so these, underground fungi, they are for the most part do not forcibly discharge spores. And so that's so a normal above ground mushroom will forcibly discharge a spore. So it's either shot off the basidia or shot out of the asci, depending on what group it's in. When truffles or hypogeous fungi rely on something coming along, smelling them, digging them up, and eating them to spread the spores. Because the way you have to picture, here's the ball of spores that aren't going to go anywhere unless something comes along and digs them up and eats them. And so that's it's a disadvantage that if nothing eats you, you're not going to spread. So the ones that don't get eaten are going to die out rather quickly, and the ones that everybody loves eating, everybody being the squirrels mostly, will spread all over the place. And so it's this, what they're doing is they're emitting odors. Most truffles have strong odors. Some are really pleasant smelling, some are really unpleasant smelling. And we'll get more into that as we go along. But when they're mature, they give off those odors and they attract, in this country, mostly rodents that come dig them up and eat them. And then if the squirrel poops in the woods, it poops out the truffle spores, and they can germinate and grow, at a new site. So it's quite an elaborate way of getting your spores around. Most of them are ectomycorrhizal, and so that's the symbiotic relationship where they are growing symbiotically with the tree, and a mutualistic relationship, exchange of nutrients going on there. And there's truffle-like fungi in a lot of different lineages. So name the group of fungi. There's probably there's almost most definitely a truffle relative of that somewhere in the world. So one thing I want to show you here is the whole progression underground, because I mean they didn't start off as being truffles. They have ended up going there, repeatedly, across evolutionary times, repeatedly going underground. And so this is just an example of one of the red port boletes in California, Swillellus amygdalinus, a liver bolete, and a gastrobolete. Gastrobolete is kind of the catch-all name for these Secotioid boletes, which are these boletes that most of them don't forcibly discharge spores. Some still do, but they've just become a little really convoluted mess. If you look at the pores and tubes on here, they're kind of like all fused together. And the same thing happening with this Lactarius pallidioolivaceus, with this progression towards these two species genetically are almost identical, but they have but if you look at the gills on there, they're no longer individual blade-like gills. It's more this convoluted mass of tissue where the spores are produced, and they're not on this one, they're still forcibly discharged, but by the time you get here, so here's a bolete that's gone mostly underground. This is one of the only photographs in here that's not of a California truffle. I don't have very many. I don't have Chamomixia. I've never seen it in California. But this is a New Zealand Ross beavera, which is really closely related to Lexinops. Blue staining on them. They're a truffle-like bolete. And then what used to be called Zelleromyces, but it's really a truffle form of Lactarius. And you can see it still goes up the latex there, the milk on it. So as they continue going underground, or more convoluted, they lose that ability to forcibly discharge the spores. Something has to come along and eat them to spread the spores. And it appears in evolutionary sense, once you lose that ability to discharge those spores, you don't gain it back. So once you become a truffle, if you don't want to be a truffle anymore, you want to be, you want to come back above ground, there are ways to do that. But when you're below ground and you want to spread your spores around, you have to emit an odor that in North America, it's rodents, squirrels, chipmunks, a lot of mammals eat them. Their deer certainly love certain species of truffles. They have to smell them and dig them up and eat them for those spores to spread around. And places like Australia, there's a lot of marsupials that are doing it. Some of the bandicoots and potoroos, in season, up to 70% of their diet is these hypogeous fungi. And New Zealand, they have a lot of these Secotioid fruit bodies that are colorful and above ground. And the hypothesis is that birds in New Zealand have spread them because they didn't have, they didn't have land mammals there besides a couple bat species. So, but they have quite a few of these, truffle-like fungi. There are some that have come back above ground, and these are stinkhorn relatives. And these are a few more from New Zealand, but we have a lot of the hypogeous, Hystorangiaceae, these stinkhorn truffles, a lot of those in Western North America. But these are ones that that are back above ground, and so like the basal clade for stinkhorns is this thing Pheo beta alba, which is a New Zealand stinkhorn truffle. This part here, the top of the grub is above ground, and the spore mass, the gleba, is below ground. And so the thought is that it looks like something appealing that a bird would want to come along and eat, and in the process would eat the spores and spread the spores. Now, the more elaborate stinkhorns are the ones that come above ground, have a really smelly spore mass, the gleba on them, and then that spreads the spores. Flies and stuff will spread those spores. So when you're out there and you want to find truffles, of course you hear about the dogs, or pigs, the whole fairy tale of pigs finding truffles. They're mostly found by dogs, for at least the culinary ones. If you want to train an animal, pick a squirrel. They're so much better. But since I haven't met anybody who has their own personal truffle squirrel, all you have to do is look for the signs where they dug them up and eaten them. And so one thing that Kaz has told me repeatedly is, if you're looking for truffles, you have to stop looking for above ground fungi. And it really is true. You just have to change your whole mindset on what you're looking for in the woods, because you are not looking for these fleshy above ground fungi. You're looking for these small signs in the woods, like this little teeny hole here. This is like a smorgasbord screaming at you, there's truffles on the ground here. Or stuff like this, where there's partly eaten truffles and truffle bits on top of the surface there. And these are all stuff that squirrels have done. So sometimes you have these big glaring holes. This was a gotcha area. They even left a little piece in the bottom. Just like, this is like a big giant sign saying truffle here. And then there's the more, like this. So here's this fresh dig where something came along, smelled the mature truffle, and came and dug it up. And so what I'm looking for out there in the woods is little signs like that. So here was one in coastal tan oak forest. That was the squirrel dig. And so I raked it back and ended up there's the truffle, little patch of them there, and so that there it is. I do have to mention that a lot of forests do not allow raking unless you have permits for it. So keep that in mind. Because there are a lot of places, first of all, a lot of places it's illegal to pick mushrooms, but even in places that it is legal to pick mushrooms, sometimes it's not legal to rake for them. So you have to check, regulations on where you're hunting. Here's another example. These are two very obvious dig holes where, just where the squirrel's gone down, they've smelled that little ASCO truffle there, and I dug up in this area and found 5 or 6 of them. Just another example of a truffle dig. And so if you look in here, you can see the fresh one right in the middle there, but then there's a couple other ones that have some pine needles over it. And this is a good example of that, because on the dig to the right is nice and fresh. And so that's what you're looking for mostly, because you want an area that the squirrels just were, within the last day or so, they dug that up. And you can especially, see it when it's somewhat fresh earth there. There's no needles on top of it. So if you look at this hole here, it was dug up, a few days, a week before. There's an even older one down here. And so these are the signs you're looking for. And the fresher the dig, the higher chance you have at finding something. So if we look at that dig, the process is, once I find it, I'm scraping back the duff on top, just the loose litter and the debris. A lot of truffles are in that interface zone between the mineral soil and the topsoil. And little signs you can look for are like these mycelial rhizomorphs in here. You can see that white tissue there. A lot of them have, that rhizomorphic mat in the areas of fruiting. Skip to the next one. Here's another example. You can see right here is the dig, and it's a really fresh one. There's still like fresh earth on top of it. So what I do is I go to that hole and I stick my finger in there and check the depth of it, because that's, if they dug the truffle up from 3 inches down, that's gonna be about where you're looking for it. Scraping back the top duff layer, you do want to look through that, but then quickly, below that is where you start finding them. And so usually where they've dug up one, there's usually be more in the area. That's what you try not to do, is hit your truffles with the truffle rake and break them. And then you also want to break apart clumps of soil, because there's a bunch of species that the peridium, the wall of the truffle had this mycelium that bind to the soil, so it'll just look like a clump of dirt. And so when you have these, these little clumps of dirt, you want to break them apart with your fingers and make sure there's no mushroom in there, because there's especially stuff like the Lapomyces, they just bind the soil on the edges. So a couple of letter signs to look for. Here's little bits of truffles that squirrels ate and left little bits behind. So when you're walking around the woods, you want to look for stuff like that. Sometimes you can find like little squirrel catches where they store these truffles, and just like they store acorns, like the end of hollow out logs and stuff, or they eat them on top of the logs and they get scared off halfway through and they drop it. So you want to be on the lookout for this. One of the rarest truffles I've ever found was something that was left on the end of a log. This is another sign of, this is just like half-eaten rhizopogon. There's sometimes in rhizopogon are fruiting, there's so many of them that squirrels just don't want to eat them, I think, or they just get full and leave big bits there. So that's another way of finding them. And then occasionally there's a bunch of species that are erupting, especially in hard-packed soils. So if you're on campgrounds or along trails or some kind of, you can find these things pushing up out of the soil, or just notice bumps in the soil. So if you see like a little mound or a bump or a crack in the soil, it's a spot you should check. And so these are spots that you wouldn't necessarily check when you're looking for above ground mushrooms, but as soon as you're looking for hypogeous fungi, you really have to start checking these areas. So moving on, I'm just going to mention a couple of the early people who worked on truffles. And one, H.W. Harkness was someone who was born not too far from where I live in Massachusetts, about 30 miles away, and went out to California for the gold rush, became a physician, and then became a real estate developer and earned tons of money from that, and then decided he wanted to study fungi, and especially truffles, and so spent the latter half of his life doing that, and collected tons of truffles from California, and did the first the first works on North American truffles, so where he posted California hypogeous fungi, which had 108 species in it, 55 of which were new to science at the time. So I mean, it kind of like, it was the start of, you know, the whole like Agaricales to California. This was the first California-wide monograph. The next one is Helen Gilkey, who was, she did lots of, she worked on ASCO truffles, kind of like at the same time that Zeller was working on Basidiomycete truffles. So, you know, from, you know, early to mid 1900s. She was the first woman to get a PhD in botany at UC Berkeley. In 1915, her dissertation was a revision of the Tuberalis of California, which she looked at Harkness's collections and a bunch of more collections, and did that. And then in 39 did Tuberalis in North America, which, I mean, these are really useful publications, even, you know, still. Post that, I mean, so, I mean, there was like I mentioned, Zeller, Jim Trappy, who's the godfather of truffling up in Corvallis. You know, he started there in the 50s and still going. I mean, the amount of work he's done is mind-boggling. And then, you know, Michael Castellano is done tons of work, and he has a whole bunch of stuff coming out soon, kind of like monographs and revisions to some major groups of Western North American truffles, and lots of other people in between. So I'm going to run through some of the more the groups of truffles, highlight some of the more common things you can find in California. Before I do that, I just want to mention some terms that I'll probably use repeatedly and may not realize I'm saying them, and you may not know what they are. So some of that is the peridium, which is the exterior skin of the truffle. So that, you know, the peridium encloses the gleba, which is the spore-bearing mass. The gleba can be, you know, it can be like, you know, rubbery or gelatinous or powdery. You know, a lot of different glebas. The columella is usually like it's veins of tissue that run through the gleba. In some species, it's almost like a stalk going up through the middle of it. And then locules are the chambers inside the gleba. If you look closely there, you can see it has like kind of like irregular small locules. Rhizomorphs are the, you know, the threads or cords on the outside edges. So in Russellales, you have, you know, like above ground, you have a lot of below ground Russula. And so they were put into separate genera depending on, you know, if they were fully hypogeous or if they were, you know, semi, you know, Secotioid, so they still had, like, you know, the cap and the stem. They're macroanides, you know, Gymnomyces completely hypogeous. Same deal with Lactarius. You know, so Zelleromyces are, you know, I showed you a picture earlier, one of, you know, just completely underground. They've pretty much all been transferred to the, you know, Russula and Lactarius now. They've evolved repeatedly within Russula and Lactarius, so it's something that, you know, that different lineages within Russula have gone underground. So it's gone underground multiple times, and they're spread out throughout Russula. So for field sense, it's a lot easier to call something a Zelleromyces and communicate that. But on a you know, on an actually taxonomy sense, it makes more sense to call it a Lactarius. And then albatrolaceae, there's Leucogaster, Leucofleps, which are both really common. So looking at a Russula, this is a Russula that, if you look at it, it's mostly the gleba. It's mostly a spore-bearing mass. It still has a little stipe left there, and a little gap on the underside. And then, you know, the skin, you know, the cap cuticle, you know, is peridium in this case, almost completely encases the spore mass on this mushroom. So here you have something, here's a Russula that's, somewhat pseucotioid. This was one that was growing in Jackson State Forest, occasionally poking above the duff, but most of them were buried. So it's probably mostly relying on stuff eating it to spread the spores. That's how I found it, because something had come and dug it up. But you can see the gills are just starting to be deformed on it. You have other ones that still have a fairly well-defined stipe, running right through, or other ones that just, just had this little teeny blip of a stipe left to it. And there's still more that don't even have that. It's just a ball. And so, these are all Russula. Like above ground Russula, below ground Russula are next to impossible to identify. So if you look at your Blitz observations of Russulas, and if you have a species name to them, and I came and knocked them all down to genus, I'm sorry, it's about all you can do without a sequence on Russula. Lactarius. Lactarius crassus is a fairly common one in with true fir in the drier forest, so Cascades in Sierra Nevada. It's in the Lactarius scribiculatus group, but you can see the gills are completely deformed and often covered with the tissue on this mushroom. It still gives like scant white latex. And then Zelleromyces, these almost completely pseucotioid ones. This was actually one that Joanne Olson found at Redwood National Park. It's one that is often above ground, or just it's showing the surface above ground, and it's a lot more orange and fresh. She found it like a couple weeks before I got there and saved it for me. But I think Scott emailed me photos of this earlier this year, so it was something that comes out fairly early in the mushroom season. But orange when it starts. When you cut it and it's fresh, has white latex. It'll bleed white latex, and the spores look identical to a Lactarius spore. Leucogaster, it's another one that people commonly see because it's emergent often. It grows kind of like high up in the duff layer and gets exposed quite often. It starts off whitish, but becomes orangish red color if exposed, especially. And then if you cut it in half, it has these open locules full with with this sticky gel-like texture. Leucogaster citrinus is another fairly common one, both coastal and in montane regions. And this is just a leucogaster spore. They're these round spores that are amyloid, so they turn bluish black in Meltzer's reagent and iodine reagent, and they kind of have like that ornamentation and then this covering over that ornamentation. And I should mention now, my interest is not in eating mushrooms, and especially not in eating truffles. There are desirable edible truffles. However, it's about the same percentage as it is for above ground stuff. There maybe 10% of them are good edibles. Most truffles are edible. Remember, this is something that has evolved to be eaten. So if it was toxic, it's not a good plan to be poisonous and have something eat you and die from eating you. But then you have to remember that squirrels have different digestive tracts than we do. So, but most truffles appear to be non-toxic. Leucofleps, I will mention the edible ones when we get to them. But yeah, I mean, like I said, it's my interest in them is certainly diversity. And I did throw in the more desirable edible ones at the end. So you do have to sit through the whole talk to get to them. Leucopleps is a really common one. It's kind of like this squishy textured, misshapen truffle that's white, sometimes be a little sticky juice when you cut it. Ones are going to have these amyloid spores with like that, the thick wall, the double walls to them. Truffle spores are some of the most amazing spores to look at. If you have any interest in micro characters of fungi and spores, just get into truffles for the spores. With Cortinarius, we don't have very many true like truffle forms of Cortinarius in Western North America, but we have a group of these thick-veiled species. You see with the really strong, persistent veil, and they often grow underground. And then there's these group of these Secotioid species. So you can see the gill mass on that. It's just this one convoluted mess, one convoluted gleba. They don't open up, they don't discharge spores. But Cortinarius pinguis is a very common species, especially in Pacific Northwest, Cascade habitat. You get up into the higher elevation, hemlock, fir forest, and it seems to fruit late spring through fall. Just any time you get rain in the summer, it comes up in mass. Just a really common Cortinarius. I will mention that this is this collection was Shannon Adams' first ever Cortinarius collection, SDA 001, 5 years ago. And since then, she's collected about 900 Cortinarius. So this was the first one that started her insanity of studying Cortinarius. Cortinarius magnifilatus is, like I said, one of these really thick veiled species. They do forcibly discharge spores, but they're typically buried underground. They have tons of spores. You can collect these things and break the veil apart, and it'll just be like this mass of spores under there. So they're probably somewhat wind dispersed, but relying on stuff usually to dig them up to get those spores out. And Cortinarius verrucosporus is another one in this group. You can see that persistent veil, but it does tear as it gets older. There's a few different species in this group. A couple of them, 2 of them are described, a couple more are getting described. Hymenogasteraceae is the family that has stuff like Hebeloma, Phaeocolibia, Galerina. The truffle relative, is Hymenogaster. There's a handful of species that are difficult group to identify. They have these warty brown spores, and Hymenogaster sublaucinus is one of the most common truffles in California. It's something that if you were hunting in the spring or early summer in the Cascades of Sierra Nevada, you can pretty much find multiple times, and they often grow in these really large patches. They have a pleasing odor. It's kind of like a sweet pine pitch, almost like a sweet Christmas tree like odor. Not something that I would take a chance eating. Hymenogasteraceae has, that family has a bunch of toxic things. I'm not saying that this is toxic, I just would pass on it. And then the Hymenogastera subalpinus is one that it's fairly common with fir in the late winter, early spring. And this is one that when I found it, I get like a Reese's peanut butter cup odor, kind of like a peanut butter chocolatey odor to it. I had no desire to eat it, but it was a really interesting odor to it. As Hymenogastera was growing with oaks, so you can kind of get the idea of the look of these things. And that's about, you know, with truffles, you're getting an idea of what they look like, because your actual identification is going to have to be microscopic, because the spore difference on them is, you know, even though two may look identical macroscopically, spores will, you know, sometimes look completely different. In Bolitales, there's a bunch of different genera. A bunch of these I have not found. Partly because I just haven't been looking for truffles long enough, and then I haven't been in California at the right times of year. A lot of these bolete stuff will fruit kind of like in the early or late part of the season, so you need early rains or late rains. Like, I mean, Santa Cruz County, like, so it's kind of like, you know, the southern end of the Redwood Coast Range. If you want to find truffles there, you should be looking March, April, May, even into June. So, you know, after the rains, because a lot of these things are underground. So that's a big advantage of being a hypogeous fungus. Kind of like the, you know, one of the reasons why you're underground is because you are in these difficult environments, so dry environments where, you know, things get dry above ground, and you go below ground, you have that ability to hold, you know, retain moisture, and just slowly mature. And when you're finally mature, put off these odors and something will come along and eat you. So you do find a lot of truffles in the drier habitats, but there's plenty on the North Coast. But, you know, diversity of them, like in the Southern Cascades, just Shasta area, I mean, there's tons of truffles over in that area. So Gastroboletus is kind of like this catch-all for this group of these deformed boletes. And so this one, Boletus subalpinus, is fairly closely related to Porcini, to king boletes, queen boletes. And, you know, so it's one of the true Boletus. There's other gastroboletes, like Gastroboletus vividus is closely related to Boletus smithii. For Xericomelus, we had, you know, Gastroboletus xericomoides was genetically matched, you know, Xericomelus amylosporus. So these above ground, you know, normal shaped fruit bodies, boletes, genetically are very close to some of these gastroboletes. And in some cases, the same thing. And so there, you know, there's also Crogomphus, Crogomphus albipes, which is a you know, a Secotioid below ground Crogomphus, where you can see, you know, once again that the gills are just this, you know, glebal mass, this spore-bearing mass of tissue. And so this was one that, this is fairly common, but it went 30 years of nobody finding it. But then I revisited a few known sites of it and found it repeatedly. So it's just a case of nobody looking for it for 30 years. So melanogaster are a group of truffles that a lot of them have like a brownish peridium or dark colored peridium, and then they have this sticky black gleba with marbled with those pale veins. They'll be whitish or yellowish veins there. They're kind of gross looking and cool at the same time. There is a revision on the Western North American species that's close to being finished. You know, there'll be a bunch of new names for stuff, new species, especially down in Southern California. There's quite a bit of diversity of truffles in Southern California. You know, that kind of that drier habitat. But you know, these are fairly common. I've seen them on the Humboldt Coast before. This collection was growing in Berkeley under a Cedrus deodora. I don't remember the common name for that, Atlas cedar. And there was literally thousands of these. And so I gave a bunch of these to people to try. Some people really liked them, and some people thought they tasted like used motor oil. kind of, so some people got a really spicy herbal odor from it, when other people got like dirty oil, asphalt, coal like odor from them. And I think one of the tricks of these is the fresher the better, because the odor seems to become stronger and more pleasant as they age. And this was actually one that I dried a bunch of it for specimens, and absolutely stunk up the house that I was staying at with this truffle. I tried to make up for it by drying a whole bunch of candy caps later. But you can see the gleba on this is these stinky black sticky black pockets with that paler marbled veins. This is the one that I found on the northern coast of California, grown with Sitka spruce and western hemlock. Really dark gleba on it. Alder truffles, Alpova, this is really common in Humboldt County. Pretty much all you have to do is go under some alder trees, look for a dig, or just look around, scrape back some leaves, and you'll see it. I don't think it's a desirable edible. It doesn't have, I'd never had much of an odor from these. A lot of what you're doing with truffles is just from, you're using the essence of the odor for your flavoring, not the actual taste of the mushroom. Shad can tell you about eating mushrooms. It's not my thing. Alpova trapii is one that grows with conifers. You would kind of like that sticky yellow gleba. Rhizopogon, and chances are if you find a truffle, it's going to be a rhizopogon. They are so common. They are everywhere. They are off. There's a few species that are often emergent or eruptant, so you're going to see them without looking for them. As a group, they're fairly easy to tell because they're rubbery. Pogies bounce, so you can drop it on the floor and bounce it like a rubber ball. They often have rhizomorph ball, which is easier to see on this one. You can see all these threads on the peridium. So they have the threads on the rhizomorphs along the outside edge usually. They're really closely related to Swillus and Gomphidius crogomphus, kind of like sister group to Swillus. And in that case, they are, especially with Pinaceae, so you can find them, a lot of them with pine and Douglas fir. There's a few species with true firs as well. In terms of IDing them to species, it is next to impossible. I know that some people would say you can do it, but other people say you can't do it without sequencing them. There's slight differences in spores. They're one of the few truffles that don't have ornamented thick-walled spores. They just have these little teeny sausage-like spores. But these spores can last a long time in the soil and still be good. So things you want to look for is staining of the peridium, the KOH reaction on the peridium. Some will go blue or green. You can see on the peridium of some of these, it stains pink or red or grayish dark color. You want to look for rhizomorphs on them. But like I said, you usually can't get them past genus, which should be good enough for most people. You can eat them, but it's not like as you think truffles as being. They're not very aromatic. They're more like like a potato to stick in a stew. And I've had a couple of them now, and they weren't offensive. I guess that's, for me, that's saying a lot for a mushroom. But yeah, you can use them as just like chop them up and throw them into a stew, like I said, like you would do a potato. Rhizopogon occidentalis, if you were to go dig under your shore pine in your yard or around, you would probably find hundreds of these. They are literally under every single Pinus catorta out there. They're really common. They grow a lot of different pines. They're kind of like, they're yellowish when young. They'll develop reddish color as they get older, and they have tons of those rhizomorphs like encasing the peridium on it. Rhizopogon parksii group is a group of the darkly colored ones. There's a bunch of different species in this, vinicolored parksii group, but these are Douglas fir associates. But another group that you're probably going to see these fairly often because they're often near the surface or erupting. But if you're digging, you'll definitely see them under Douglas fir. This is another Rhizopogon occidentalis down here in the lower left. You can see kind of like these exposed areas. So that was above ground. You develop those reddish colors. But then there's some of these like pink staining ones in the the rubescence group. But like I said, there's a lot of different species of Rhizopogon. And most of them have, a pale to dark olive gleba when mature. And the gleba is like a really dense sponge, a rubbery sponge. Yet more. Those are probably all different species. And then another one that's really similar to Rhizopogon. This was actually one that has come into the Mycoblitz. I believe as Michelle Torres Grant had an observation of this really early on of the trunk columella citrina. And this is one that, it's at the surface quite often. But if you're digging, you find this thing all the time. And it's really distinct because of those yellowish colors and then the like grayish olive gleba with the columella, those veins that are running through it. So the Hysterangiaceae are these the basal stinkhorns. They are really dense, firm texture to them, firm rubbery texture often. Many of them have that olive colored gleba, and many of them have that translucent columella that runs through it. The peridium on them, the skin on them, can be really thin to somewhat thick, and it peels on a lot of them. So that's one feature that, if your Hysterangium peels or not, it puts it into different groups. And there are some really bad smelling ones in this group. Kind of like rotten eggs or, rotten meat, feces. I mean, some really unpleasant odors. A close-up of a Hysterangium. Peridium staining is important on this group. What if it's staining pink or red or brown or purple? Important to note. Some more examples. You can see on this one how that peridium is flaking really easily, and that would just peel right off. You can see where it's been cut. It was tough to get this to not peel when cutting it. And this is a group that there's going to be a bunch of new genera in this group. Teresa LaBelle and Michael Castellano and a few others are working on revising this whole group. So there's a bunch of different species and different genera that are all lumped together right now. But you can see the gleba on this with the translucent columella, and of course the staining. This one stains orangish brown where it's touched. And this thing reeked of rotten eggs. Trapea darkeri is a really common one, both coastal and montane. It's another really putrid odor, and fairly easy to recognize. If you look at the edges, it's a false peridium. It's really just a pale line of the gleba. So those glebal chambers continue right to the edge. So when you're looking at it, it's not, that white line's not a peridium, it's just a white gleba. So it's a, it looks, it's the same texture as the gleba, but and the peridium just on the very edge. So it's the easiest way to recognize Trapeoidicerae, which I said it's common. Trapeophilipsii is a rare one that I think there's only like 15 records of it or so. It stains pink really quickly when you handle it. And you see that it's, even though it looks water, it's a solid translucent gel inside of that, in the middle of this one. One of my favorite groups is Gattieria. Extremely abundant, extremely diverse. They're very closely related to Ramaria. So the coral fungi, they're often rubbery textures. There's a few that are a little bit more brittle textured. You can see that they most often have a columella, which is kind of that that stipe remnants, so just those veins or clump at the bottom, sometimes just like a little bump at the bottom there. There's quite a few really unpleasant smelling Gattieria. Gattieria monticola is the catch-all name for all of them, but there's 40 or so species in California, I think. This is one that there should be a monograph for Western North American Gattieria fairly soon. But when you start looking, you will start noticing the difference. This is one that does not have a peridium. So you can see, just what I want you to imagine here is to take a coral mushroom, Ramaria, and squish it up into a ball. And so what you're getting is you still have the structure, you still have that stem and all those branches, but now it's in this ball and it's gone underground. And then a bunch of them have gotten that, that skin over the whole thing, the peridium over the whole thing, added protection, I guess. But you have a lot of different species of these. And it's not surprising when you go into the California mountains, it's dry. And if you're Ramaria, deer eat you all the time. I mean, how many times have you seen Ramaria that aren't deer munched? So they have, the advantage of going underground. This is a close-up of the Gattieria gleba. And once again, you can picture kind of like all these Ramaria branches, and then the spores being produced in those locules, in those open pockets. And so a lot of Gattieria all mature to about the same color, and kind of like that ochre pinkish color. And then Gattieria spores are absolutely amazing to look at under the scope. So these like I said, lots of different species, so lots of different shaped spores. These little wing spores, the longitudinal striate spores. I mean, you can see these, and then they have like those little wings on them as well. But it's something, definitely find Gattieria, scope them. You'll have so much fun doing it. And then in terms of, like I said, lots of different species out there. They're really common. A bunch of them you can probably, you can sort of tell some of the groups macroscopically, but you really have to put them under a scope to tell them apart. And once you do, you'll see how different these spores look. I just grabbed, like 4 different collections out of my, collections and threw them under the scope just randomly, and there were 4 different species. But you can see they all follow that similar, the colors are all about the same. The gleba can be, there's some that have really large locules, large openings, some that are really tight, densely packed and more rubbery. Couple paler species, couple darker species. The peridium color matters if it's really pale and white versus dark, more purplish color. My favorite one is the banana god, And so this is an undescribed species that grows under high elevation red fir forest. And when you first smell it, it smells like caramel bananas. It is absolutely unmistakable. You know, and this is one that I've given to lots of different people who are, you know, not mushroom people, just to smell, and they like, and you know, ask them to name the smell, and most people get bananas right away. The problem is, after your second or third sniff, you start getting like acetone odor, really chemical odor to it, and it's not very pleasant. And so this is one of the few truffles I have eaten. I flavored yogurt with it. So I just took one of these truffles and put it into, you know, I put a bowl of yogurt in the fridge. I put a truffle on the plate next to it and covered it up overnight, and then tasted it the next morning. And it was so bad because none of the banana flavor got in there. It was all that wretched chemical odor. And then another Gaultheria story I have is one of the times I was driving from California back to Massachusetts, and I had a bunch of truffle collections, and there was a couple Gaultheria in there, and I just like, I was mushrooming the day before, and then I left to drive across country, and I was like, oh, I'll deal with these in a day or two. By the time I got to Nebraska, I was so sick of smelling the Gaultheria that I just like stopped the car and took it and threw it on the side of the road. And it's like, I am not going to deal with this. It was just, that smell just was so bad. A couple other ones that I want to quickly mention: Distancia, another rare truffle, really neat things. Keep an eye out for them. They have white peridium that'll stain pink and purple. There's a few different species. Sedecula is this weird thing that grows in high elevation forest that kind of like this top shape with dark powdery gleba with these like thick rhizomorphs coming through it. This here is a Lepiota. So yeah, it's a Lepiota truffle. But they're really rare. This is only the second collection of it. There's another one that's another species from Oregon. It's not described yet. And these are geastrum truffles. So kind of like, you know, these Earth Star like truffles. And then another one worth mentioning is the Foliota. This is a common one, you know, Secotioid, not truffle form, on especially like fir, another higher elevation fir forest, Foliota that fruits on that, you know, the edges melting snowbanks. It smells like wintergreen toilet bowl cleaner. Some of the older mushroomers in California say it smells like bubble gum, but I don't know what they were eating for bubble gum 50 years ago. But it's straight up toilet bowl cleaner. And Dogny is a zygomycete. All the ones I've showed you so far are basidiomycetes. There are a few zygo truffles. This one's fairly common in pine forest in California. And then we'll move on to the ASCO truffles. So these are a lot of the edible ones are, you know, the desirable edible ones are the Ascomycete truffles. You know, it's where you get the tuber species. But we'll start with the Lapomyces. Lapomyces are probably, I mean, it's probably, if you had to go out, you know, any day of the year, and I say you had a chance of finding a truffle, you would find the Lapomyces. You know, for me here in Massachusetts, I can literally go out anytime I can dig up the ground and find one. And for some areas out west, that's true as well. Lots of undescribed species still that's being worked on. Most of them have a rather thick and tough peridium, so it's a tough woody skin, and then that powdery gleba. You have these globose ornamented spores. The Lapomyces granulatus group, it's everywhere. I mean, it literally anywhere you go, and you know, conifer forest out west, you can probably dig this thing up. But you can see the, excuse me, the rather thick peridium, and then that dark powdery spore mass. Elaphomyces miracatus group is a group that has the marbled peridium. So when you do a cross section, it's got that marbled look to it. This one's really young, so it doesn't, so it's still white inside. But then there's a bunch of other ones, fairly common in California. I would not eat an Elaphomyces. Some people do, but I would stay away from them. Geopora, this is one that you can eat. Very common, often near the surface, so it's noticed often. They're called the hairy truffles. The peridium will be fuzzy when they're fresh, and then it's just this convoluted mass, these folds, that make up the gleba on it. It's reminiscent of morels. I mean, it's a flavorful, crunchy texture. I see a lot of it like the year or 2 years following forest fires, burnt areas, both spring and fall, but it's plenty of it in unburnt forest as well. So when you find a truffle that has, the hairy exterior and that convoluted, mass-like interior, chances are you have a geopora. There's more cup-like geopora as well. This is one that was down in Irvine, California, that was mostly buried, but a little bit of it above ground. But once again, the hairy outside, just a simple cup. There's a bunch of these that are convoluted, small, ASCO truffles. They're difficult to find. They're not beginner truffles because typically they don't have, there's not big digs for them. They're kind of like in the duff. They often bind to material, but they're really neat. I mean, I don't know about edibility on them. Hypnotria, I don't see this very often. It's kind of like, there's once again, more like this convoluted brain mass, kind of like, that distinct color to it. This one smelled very cheesy, like strong ripe cheese odor. Genevia is I would say it's not uncommon, fairly common. I mean, if I'm out in California, I'll see it 2 or 3 times a year in the springtime. But it's tiny. It's about 1 centimeter across when it's big, so it's easily overlooked. But often when you find one, you find a whole bunch. Barcia is one I haven't. This is the only time I found it. It's similar to Balsamia. Balsamia are fairly common. Not much of an odor to them. I mean, I would imagine they're edible, but there wouldn't, there's not much aromatics coming from these things. So Balsamia, this group was recently worked on, and there's a bunch of new species there, but they have that marbled interior that's similar to the tuber species, but the exterior on them is often like warded or bumpy. And the spores are quite different. They're more like normal spores compared to tuber spores. But there's a bunch of different species of Balsamia. Chloromyces is an uncommon one that's in red fir forest, and this has some of the neatest spores. It's a really pleasing odor as well. It's probably a pretty good edible one. I've only found it a handful of times and have always saved the specimens for herbarium, not for eating. But the spores on it look like little golf balls with alien eyes. They're super weird spores. And then the last group is the tuber species, which have, they're firm, really dense, irregular shape, lobe fruit bodies. They have that marbled gleba to them. Many of them have really strong, pleasant odors. If you like the truffle odor. I mean, it's a really distinct odor. There's no easy way to describe a truffle odor than it smells like, it smells like a tuber. They have a, just a really distinct odor that they have. And they have some of the coolest spores. But this is just a close up of that marble gleba. You can see it's solid, but then it has these, these paler lines that kind of like, vein through it. And so that's what you're looking for. If you find a truffle and it doesn't have that, then you probably do not have a tuber. Tuber candidum is a common one with oak. It just does not have an odor to it. So it's one that you could eat, but once again, it's going to be like a potato truffle. It's not going to be your strong odor truffles. They have these spiny spores without a reticulum. But there's a bunch of different species with the oaks. This is just, especially like blue oak, kind of like these habitats you would never expect mushrooms in. When you start digging under them, you'll start finding quite a few different ASCO truffles. I believe this is Tuber californicum. I haven't scoped it yet. But once again, not a strong odor one. The Oregon white truffle, Tuber oregonensis, this is when you start getting into the highly desirable ones, the ones that are commercially harvested in Pacific Northwest. They do occur in California. This was from Point Reyes. But this is, the whole Oregon white truffle group is of 4 different species. The 2 common ones are oregonensis, which these are the spores from it. And if you look, they have spines coming off these honeycomb-like pattern, and they're in these little, these little sac-like asci. And then this is another one of the Oregon white truffle, Tuber globosum. This is probably the most common one. And this, I mean, it's fairly common in California. And this is, when mature, this is like the highly prized edible one in Western North America. Some would say it's on par with the European stuff, but it's not. There was issues with just like general raking and lots of immature fruit bodies on the market, so it got a bad rap because of that. Now that there's more and more dog harvested ones are only picking mature ones, you definitely only want to eat the mature ones, because an immature one's not going to have an odor, and so and not the flavor as well. So keep your eye out for this one, because I mean, it can get pretty big. These were tennis ball size. And Belosporium is another one in this group. So if you want to go find truffles, make friends with the squirrels, because they will find them for you. It's a lot easier to have a pet squirrel than a pet pig. So I hope you can go out there and look for these things, yeah, I mean, open your eyes to the world that you don't see all the time. So with that, I can start taking questions.
Okay, good, because you have a lot of them. Okay, so we had a few questions about habitat, a few about different types of forest habitat that you would want to look in, and then just habitat in general, where to go find them.
So anywhere you find mushrooms, you're going to find truffles. Pretty much every single habitat in California, outside of the driest deserts and destroyed farmland in Central Valley, is going to have truffles. So I mean, there's certainly better places. If you want to find diversity of truffles, go to the Southern Cascades, Mount Shasta area in the spring or fall. In those drier, kind of like those drier habitats, you're going to have a higher percentage of the fungi underground. If you want to find the tuber species, the edible, Tuber globosum group, go to Young Douglas Fir Forest. And when you get up in Oregon to those Douglas fir plantations, you know, those 25, 30-year-old trees, that's the habitat it loves.
And then would you recommend covering a lot of ground or focusing in one area?
It depends. It depends what your goal is. Like when I'm hunting in the mountains for truffles, I will give one spot like 30 minutes or so. If you're not finding them there, move on. You know, just continue moving on to the next spot until you start finding them. Because I mean, there'll be a period that they're fruiting and a period they're not. So in anywhere you can get a gradient in elevation, you'll go up or down. If you're not finding them in coastal, pretty much if you're not seeing above ground fungi, you're probably not going to see truffles. Truffles are usually out a little bit later than, you know, at least they're mature, and in that way, the way you're going to find them, because something's dug them up later than the above ground stuff.
Then there's a few questions about spores. Do the hypogeous forms contain more spores than above ground forms?
I don't know. I mean, the above ground form can produce spores over a longer period. I don't know if anybody's actually counted the number of spores that come out of a guild mushroom, you know, and then the hypogeous close relative to it. That can be your project. I mean, just whoever asked that question, it's what you're going to figure out.
And then are truffle spores more interesting, I'm assuming looking, because of adaptations needed for surviving the GI tract of animals?
Probably, because all that, you know, all that ornamentation or thicker walls is extra protection. So if you're passing through, you know, a mammal, you want some protection. But then rhizopogon, how do they get away with it? I mean, how are those little thin-walled sausages just like, you know, squeak through and you know, become everywhere and really diverse? But yeah, like stuff like the Xericomellis truffles. You know, there's a couple truffle forms of Xericomellis, and they have completely different spore morphology than the bolete forms.
And then you got a question from Shannon. You say, we do not have many I keep saying this wrong, hypogeous cortinarius in North America, with a lot of confidence. Have they been studied enough to know this? And which also above ground genera are most common?
Yeah, I would say that, I mean, because truffles have been fairly well studied. They've been better studied than above ground stuff. Just because you know, the Corvallis crew has done, you know, amazing work in Pacific Northwest on getting out there all the time and looking for these things. And there's probably a higher percentage of truffle people in the Northwest than mushroom people, in terms of, you know, people actually doing work on serious work on, you know, taxonomy of truffles versus mushrooms. There are quite a few hypogeous cortinarius in Australia, and lots of truffle forms of cortinarius in Australia. But for North America, I don't know of any true truffle form. There's, you know, there's a handful of Secotioid forms.
Are all truffles mycorrhizal with trees, or are any with shrubs?
There's only so much shrubs. Trying to think of, like, there are ECM shrubs. So, you know, Archistaphylos, the manzanitas. There's some truffles with them, and, you know, there's a lot of shrub forms of manzanita. But yeah, I mean, there's like cistus or dryas, you know, there's truffles with those as well. Or dwarf birch, dwarf willows.
Which I suppose this also relates to another question, and I'm going to maybe say this genus name wrong. What does Genebia associate with? Possibly manzanita?
There's a bunch with oaks and there's a bunch with conifers, but possibly with manzanita as well. I think that there's a handful of species from Southern California I haven't seen. The one I found that, yeah, I mean, I found some in the Sierra that, yeah, there's pines around, but there's also, you know, there's Archistaphylos in a lot of places too. So it's just tough to pinpoint sometimes.
So what is the difference / relationship between rhizomorphs and mycelium?
Rhizomorphs is just a term for a clump, a thread-like clump of mycelium. So it's, you know, you could call it mycelial rhizomorphs if you want. It's just a thread of mycelium, you know, aggregated thread of mycelium. But why some things have it, in terms of like the trapea of the really big rhizomorphs, that's a saprophytic truffle. So that probably has something to do with it. But for rhizopogon, yeah, I mean, what advantage does that have? I don't know.
Then we have a question about comparing to Italian species. So why so shallow here when they are much deeper in Tuscany and Piedmont? And thanks for the great talk.
There's quite a few species here that are quite deep as well. Tubers are usually pretty deep. You know, both here and, you know, so the Italian truffles are, you know, they're talking about tuber species whenever Italian truffles are mentioned. But tubers here are usually pretty deep. The little oak potatoes, Candium photos I had, those were all 8 inches down, you know, in thick clay soil. Elaphomyces will sometimes be a foot down, you know, way down in the mineral soil, you know, for those, you know, those montane species. But there's the ones that people usually find are the ones up at the surface.
So could a single species display both above ground and hypogeous phenotypes? Am I saying that wrong? I feel like I'm saying
hypogeous, yeah, that's fine. Probably not if it's fully hypogeous, but what we found with the Blitz, and we have to decide what we're doing with Gastroblitz vividus and Boleta smithii, because based on ITS, it's the same thing. One of them's a deformed, you know, non-ballistic spore thing. One of them's a normal bolete shape. So yeah, I mean, possibly.
Well,
but I would
I'm
say
just
fully
wondering,
hypogeous, no.
Have people looked at other gene regions for Gastroboletus to see if there's some other major changes, like where they wouldn't be able to mate with each other, or if they just looked at ITS?
No, there's been some multiple gene sequence. That's what we have to do with Smithii vividus stuff now.
Let's see. Do you think this form of spore dispersal evolved with the radiation of mammals, or were they common before?
No, they probably evolved, you know, co-evolved with mammals. It's why that, you know, like the New Zealand stuff, you know, a lot of it's still above ground and fairly brightly colored, because it's believed it evolved with birds there. But it appears to be hand in hand, and it's a combination of, you know, environmental pressures. Looking at the question and answer, I see that, you know, why not get eaten above ground? Why do truffles go underground? So picture the California mountains. You have snowmelt moisture, and then it gets hot and dry, and your whole summer can be really hot and dry. Well, if you're 6 inches underground, it's going to stay moist there for quite some time. So your window to come up and grow and mature, and then something comes along and eats you, it can be a month or more. If you're above ground, you're going to dry out in a day or two. And the deer are just going to come along and eat you anyway. So if you're a Ramaria, why not become a Gatiaria?
And then somebody asked, before you started talking about the banana got, they
Yeah.
found a truffle that smelled like a ripe banana, but then they clarified that they were going to say banana citrus, and that they carried it around for several days and still thought it smelled great. So if you had any ideas on what that was.
I would say leucogaster. Which when I found, I mean, I think I described the like collections as like pina colada smelling. So it's kind of, hold on, I'm just trying to share the photograph here. So my guess would be one of these leucogasters. Because they're often like, like pineapple smell or, coconut pineapple, really pleasing odors, often on the surface of the ground, so people find them. So I would keep an eye out for these with, if you have, if you're looking for that odor, those sweet fruity odors.
Are there any poisonous or toxic truffles on the West Coast that we should avoid?
Probably. Do I know what they are? No. I would avoid, like I said, I would avoid the cornarius. I would avoid the hymenogasters. I'm the wrong person to ask about edibility on fungi. I wouldn't eat Elaphomyces. I think everything else is okay, but don't take my word for it. I mean, I wouldn't eat any of them. I mean, if you want to know the truth, I find truffles all the time and I have zero interest in eating them.
Okay, we have a follow-up to the underground question. Are there tropical hypogeous fungi where drying out is not really an issue?
Yes, there are like New Zealand has hypogeous fungi, and the thought there is that it's so wet that the advantage of going underground is like, for protection as well. So rather than being above ground, being inundated nonstop and having your fruiting structure destroyed by rain and moisture, if you're underground, you can be protected. And in terms of tropical stuff, yeah, I mean, for the work that Terry Hankel's been doing in Guyana and Cameroon, he's found a number of truffles, in both places. In Cameroon, Elaphomyces are really just 3 species of Elaphomyces he found there are really common, and a couple things in Boletaceae as well. Guyana had a bunch of stuff in Boletaceae, among others.
Other than getting a pet squirrel, do you have any tips for decreasing forest floor disturbance?
Stop allowing clear cutting and logging. I mean, I know that people worry about digging up a truffle in a small area. And yeah, I mean, but you got to remember that these fungi have evolved to be disturbed, to have something come along and dig them up. If you ever look how deer dig stuff up, they are really messy. And deer dig up a lot of truffles. Squirrels are neater about it, but in terms of doing it, don't dig in the same area every single year. Replace your divots. So when you see, I scrape that duff back first, I put it in a pile, I go through the soil, I look for the truffles, and then I cover it back up. And so in a few weeks, a month, you can't really tell that somebody dug there. But yeah, when you're talking about a spot that's a foot across compared to millions of acres that are clear cut, fight bigger battles.
That's great advice. Speaking of digging, if you dig up cordyceps, will you find Elaphomyces?
If you dig up the Elaphocordyceps ones, Tilaplidium, that are on the Elaphomyces, you will. You have to be careful because you can break them off really easy. That's like the beacon. That's like the tells you that there's a Laphomyces there. But if you went and randomly dug in the forest, you would find a Laphomyces in about half those digs, I would think. I mean, just certain areas, you can literally go and randomly dig and find a Laphomyces. But the cordyceps are really easy to show them off.
All right, and then we have one more question in the Q&A. Can you dye fabric with truffles?
I don't think Alyssa's tried any. Gaultheria are closely related to Ramaria. So for those of you who saw Alyssa's talk last night, she had like, you know, all the Ramaria tests. There's no reason why Gaultheria shouldn't, because they're in the same, you know, same group, closely related to, they're in the group of Ramaria that are dye Ramarias. You know, the brightly colored Ramarias like, you know, Ariospora, Stancii, those really bright pink and red ones do not dye. It's kind of like those dull beige and yellowish ones that are the dye Ramarias. So those probably would. The Ascomycetes one probably would not, although morels do dye. She will never tell you that, but they actually do a nice blue dye. She cringed at the fact of using morels for dyes, but they work. So yeah, I would imagine some of the ASCO ones may, but I think most of them would not. But any of the dark pigmented spored ones, you could try.
And then we had a question in the YouTube before you started talking, but I didn't get to it. And it was actually about Lactarius. So are all green staining orange Lactarius considered in the Lactarius deliciosus group?
If you want to, be that broad, yes. But I would say that I don't consider Lactarius rubelacteus in the deliciosus group. So for when I use, I mean, it's personal. You can, they're all in section deliciosi, which is the section of Lactarius they're in. For the deliciosus group, I want it to be those orange latex, at least orange latex to start. Mostly green staining. Estivus doesn't really stain greenish, or just really faint green. And that's in the deliciosus group. So I would say all the orange latex ones to start, because rubelacteus is red latex to start. But if you want to call it the deliciosus group, that's fine. I'm not going to argue with you.
And then Alyssa Allen actually chimed in the YouTube and said yes to try the Gaultherias for dyes.
Yeah, so one thing about Gaultheria is you can find hundreds of them in an area. And the squirrels don't seem to like eating them either. They'll like dig them up and take one bite out of them and leave the rest. But some of these Gaultheria, I mean, just giant patches of them. So, I mean, get into the mountains in the spring, especially, and give it a try. You can try eating them too, see if you like them.
We have a very specific question about Leucangium carthusianum. I only ever read
the
these
Oregon
words.
black truffle. Yeah, it's the Oregon black truffle, and the reason why it wasn't in my talk is because I've never found it, and I didn't want to use anybody else's pictures in my talk. I need to find that mushroom, that truffle. I've looked for it. I've never looked for it with the dog. That's what I'm going to have to do. All the stuff I found is without dog assist. And so when dogs are looking for truffles, they are trained to find, they mostly find the edible truffles. They're trained for a certain, a couple odors, and they're mostly finding things with those odors. And that's the tubers and the Leucangium. There's another one that's often called the Oregon brown truffle, which is a close relative of morels. I haven't seen that one either. Those are both highly desirable edible ones. I just have not found them. I did ask somebody for a photo today for Leucangium, but I didn't get it in time. But it is something that should be in the talk. It is in California. I mean, it's not as common in California, but it's fairly common in Oregon. Once again, young Douglas fir forest. If you want the edible truffles, you go to young Doug fir.
Okay, I think that's all of the questions. I might have missed some, but I think I got them all.
Let's. Were some of the trap names, are they named after Jim Trappy? Yes, they were. There's, you know, so Trappy is named for Jim Trappy. Okay, if there's any more questions, it's your last chance. It doesn't have to be about truffles. If you have any mushroom questions really quickly.
No,
I have a
I feel
question.
like at all.
Oh.
Go for it.
Can I ask a question? So
You may, Joanne.
on our dunes here, we have truffles of various kinds, but I'm told that we don't have any squirrels on the dunes. So I wonder what mammals might be digging them up.
Any rodents, I mean, literally anything that can smell them and, you know, will consider them food. But that being said, I don't see a lot of digs on the dunes. So if you go out there and if you rake under any of those pine trees, you'll see hundreds of Rhizopogon occidentalis. So, you know, the spot in those outer dune hollows with the, you know, the little short trees, a couple of those photos came from out there. I think the photo of mushrooms of the Redwood Coast is from out there. All you have to do is pull back the duff there and there's gonna be lots of Rhizopogon. So they're out there, but they're not really being eaten out there. If you remember the picture from early on in my talk, showing the pine needles, let me just pull this up, showing the pine needles with the squirrel digs. This was on University of Washington campus. And literally under the pines, you know, there was digs everywhere. This one shows it better. Where in under one tree had about 100 digs. And so they had gotten most of these truffles. I think that if there was squirrels out in the dunes, there would probably be a scene like that underneath those pine trees as well. So you can just, every single one of these holes there was a truffle in there.
Hmm. Thank you.
You've had a ton more questions pop up now.
Okay.
Kyle wants to know what your least favorite mushroom is, and so do I.
Oh, that's a tough one. I would just say, I'm gonna get in trouble here. Inocebi. Just the whole group. Russell is probably a close second. I mean, I don't know. I don't have favorites or least favorites. I mean, I'm just happy when there's mushrooms in front of me. I used to say that about Hebeloma, that, you know, the last mushroom I would ever collect was a Hebeloma. And since I've said that, I've probably collected 400 of them, 350 of them. I don't know, a lot of them. But that's because Henry Becker was working on Hebeloma, and here was a chance to like finally, you know, collect these things and have someone work on them. So if someone came to me and said, I will work on every single Russell you collect, I will start collecting them. You know, it's one of the reasons why I collect hypogeous fungi. So yeah, I would right now, I would say Inocebi is my least favorite. Now Brandon Matheny will never talk to me again.
More about your sort of personal relationship with mushrooms. How did you first become fascinated with mushrooms, and did your parents take you shrooming?
I was 7 years old and I went on the local mushroom club walk. And I had an older brother who was a birder, and I was trying to compete with him and I couldn't, so I needed something different. So mushrooms were a good choice. And then, yeah, and before I realized it, I mean, there was you know, I got too good just to quit. And then before I realized it, it was 24/7 for my whole life. And that's what I did. And now I can't stop.
Are you aware if any work has been done on species used by flying squirrels?
Yes, there's a lot of papers done on rodents eating, you know, not just animals eating hypogeous fungi and what's in their diet. If you want, there's lots of fascinating papers on that subject, and a lot of the work done in California as well. So that's something that just Google that and start looking for them, or email me, and and I can start you down that rabbit hole. Just find one paper, read through the references, get all those. You can read for hours on it. And all you have to do is just start looking at squirrel scat and look for spores in the scat. There's a lot of work done in Australia on that as well.
Oh, that ties to two things that came up. One's in YouTube about spores and scat. Do spores have to go through a digestive tract to be viable?
Probably not.
And then the other question that relates is,
don't.
Have you made it to Australia yet to look for truffles?
I have. The problem was because I haven't mushroomed much in Australia. I've been to Australia 4 times, I believe. I can't even remember now. It's been, it's 4 different times. One of the time was intentionally not in mushroom season, so I wasn't distracted and can see other stuff. But when I was there in mushroom season, I am too distracted by above ground stuff to start looking below ground. So that's a problem still in this country as well. It's really, my truffle hunting is not, because like I said, you really have to come on, just like, if you're looking for truffles that day, you're looking for truffles and ignoring above ground stuff. Or if you're looking for above ground stuff, you're ignoring truffles. You can't do both and be good at it. So in Australia, I've been too distracted by stuff above ground, but I did a little bit of digging there. Lots of activities there. It's okay, dogs bark.
I think she stopped. Okay. Have you found, or have you heard of or seen any species of Ganoderma fruiting underground?
No. There's no polypore truffles that I know of. But there are a bunch of polypores that form underground sclerotia. You got quite a few of them. They just form these large underground sclerotia, but it's not a fruiting, it's not a spore producing structure.
Are there any truffles that are related to Cantharellus?
Good question. None that I can think of off the top of my head. There must be in Australia though, because everything's in Australia. None that I can think of right now.
And then we had a question about edibles. Could any of these edibles be preserved better than others?
Preserved in like what way? The odor on them is kind of volatile, and it goes pretty quickly. And so like I said, I'm the wrong person to ask about edibility with these things. Ask Chad, he's speaking Friday night. Ask him these questions, because he'll have answers for you. I'll just like turn my nose up and like, so one thing, last year at the Mount Pisgah mushroom show, the Truffle Society had like all these truffles sitting out there, and and you could pick up the little cup and smell them. And Elsa and I walked over there, and there's these people smelling them, and I said, "Oh my God, it's so amazing!" And we both picked it up and smelled it, and we just like, "Oh God, that's awful!" And that was our response to it. And that is, and in all honesty, that's my response to, like, if someone uses truffle oil on something, or that truffle popcorn, I find it wretched. I really can't stand that. I've had, the highly prized European truffles, and they don't do it for me. It's an acquired taste. And if you don't like the odor of it, you really won't like the flavor, because the odor of these truffles is pretty much the flavor of it.
Mm.
Speaking of acquired taste for odors, do you think matsutake may be going the way of the hypogeous, with time spent underground and the strong odor?
Yeah, it wouldn't surprise me. I mean, give it a few more million years, and stop picking it. I mean, you gotta let natural selection work its way. There's certainly fungi that you can see that happening with, in the California mountains. There's one, it's called Hygrophorus ceruleus, but it's really a Clitospebe. It's closely related to Clitospebe odora, and it's this really putrid, rancid smell to it, and almost always underneath the duff. And you can see, like, you'll see like flies going to this hole in the ground to go find this mushroom, and occasionally it's dug up and eaten. So yeah, there's probably these bunch of gilled mushrooms that are like, give them enough time, and the habitat extremes, stick it into a habitat that dries out, and give it enough time to change. It may.
This is a question that I had too when you were talking about figuring out how deep a hole was and you just put your finger down there. Do you ever worry about any other critters being in the holes when you go hunting?
In this country, no. There's plenty of better things to worry about. Like 70 million Trump voters.
Ooh.
I didn't say that out loud, did I?
Oh. Thank you, Noah. It was a great talk.
Thank you.
I'm going to
Yeah,
Thank you, Noah.
Yeah, I went back
Hey,
on
have
audio
fun,
and video.
everybody. Go out
I
Oh,
there and
would say
look for
thank you.
truffles.
There were an awful lot of questions.
Yeah, I'll be looking for truffles now.
too.
I
Make
don't
INAT
see-
observations of them. See everybody on INAT.
All
Yeah,
right,
yeah,
Noah.
they'll be there. All right, good night.